We have, given Kristen has done a lot of work, advocacy work, we wanted to invite our U.S. House of Representatives from Alabama's 5th District, Congressman Mo Brooks.
Yeah, I think it's a little bit unusual because I'm supposed to give a welcoming set of remarks and y'all have been here for a while. But in any event, welcome to Huntsville and the Tennessee Valley of the state of Alabama. It's a beautiful community. From what I understand, many of you are from around the United States and for that matter from different parts of the globe. And I hope that you're finding your visit here enjoyable. We have an old saying that I'm going to mention just very briefly. It was, I first heard it from Mayor Joe Davis back in the 1980s. He said that it's a lot easier picking tourists than cotton. So please, as our current mayor, Tommy Battle of the city of Huntsville, and Paul Finley, mayor of Madison, would say, spend a lot of money while you're here, okay? We have, of course, some unique things if you like technology. Space and Rocket Center, very close, nearby, wonderful place to go visit. If you're more into nature, we've got Cathedral Caverns that is about 30 miles to the east, and it has about 5 or 6 world record geological features in it. It's really a remarkable cave. It's paved and lit up, so it's not spelunking. It's a nice walkthrough kind of thing, but it's a beautiful place to go. And if it was the summer, then I would share with you what some of us Southern boys would be doing. We'd go over to the Bat Cave where there's an endangered species, and about 500,000 bats will fly out at dusk, okay? And it's a small area opening that they fly out from, and to stand there in that opening with all those bats is a very memorable experience. But I would add that if you choose to do that someday, make sure you bring an umbrella because all those bats just woke up, okay? On a more professional level, I very much appreciate what y'all are doing with PTEN. It's very important. Particularly for the people who are adversely affected by it, for those who get some of the consequential effects of it. By way of example, my grandfather died of cancer. My father had cancer, both prostate cancer, and I just had my surgery back in December. And unfortunately, cancer is one of the adverse effects of some of these issues that y'all are discussing today. So I do want you to know that we very much appreciate the time and dedication you are showing to try to advance a cure for this and other consequential illnesses. That's what it's going to take. Get that momentum going in the United States and also around the planet, and hopefully we'll be able to cure this disease as we've been able to miraculously cure so many others, things like polio, and the list is very, very long. The improvements in our medical knowledge and capabilities is remarkable, but this is one where we need to step up our game a little bit. So with that, thank you so much for the opportunity to be here, and I hope you enjoy your visit to the Tennessee Valley of the state of Alabama. Y'all have a good day.
Okay, back on track here. All right. Claudio Ales is the founder and president of PTEN Italia. Since 2014, Claudio has worked in management with the Center of Genetic Rare Diseases. With PTEN Italia, Claudio has created a high-level scientific committee which is affiliated with UNAMO and Eurodis. PTEN Italia is listed in Orphanet. Claudio served on the board of UNAMO and was elected EPAG representative in May 2016. In February 2017, he moved to Genturis ERN. Ales attended International Summer School for Rare Disease and Orphan Drug Registries in 2006, and the Eurodis Summer School in Barcelona in 2017. He participated in the European Conference Rare Diseases and Orphan Drug in Edinburgh and in Budapest. Claudio is a champion of the PHTS community and has a proven track record working for those who suffer from rare disease, often being invited to speak at national meetings, such as this one, for rare diseases and undiagnosed rare disease. Please welcome Claudio.
Okay, thank you. Good morning. I'm Claudio Ales, President of PTEN Italia and IPAC representative in here and Gentouris. The initial problems, the first problem for the PHTS patients, you know, the desert information, no cure, no treatments, no diagnosis delay, and loneliness or dismay and fear. The first steps for the association is to create the association. The association that began on March 2013 by the will of 3 families, me, Anna, and Mary. And the first step is the subscription on different list of association or different institutional organization. We are affiliated on Uniamo. Uniamo is the Italian Federation of Rare Disease, and affiliated on Eurordis, European Federation of Rare Disease, that has maybe 700 associations in Europe and around the world as the PTEN Foundation. I'm on board of Uniamo, the Italian Federation. The first steps is to create the scientific committee. It's very useful for us, constituted by 4 important geneticists, Italian geneticists from different regions that know the PHTS. We start with the participation in national and international meetings, conference, workshops, and school on rare disease. As Eurordis, as Teleton, and the winter school of another winter school of Uniamo. So it's very important, this participation that consent us to have an accreditation in the organism. For example, in the HERENS. HERENS is European Reference Network. Is a virtual networks involving healthcare providers across the Europe. It's very important because there is a center of coordination in one of the member states, and there is a network with different healthcare providers in Europe, in different member states. Italy has the high number, 190 healthcare providers. This is the roadmap for the patients. The patients adverse national care providers, then the HEREN, and in the HEREN we study the clinical guidelines, the research, the sharing of evidence between different care providers. In Europe there are 24 ERN different. I'm in Genturis. Genturis is genetic tumor risk syndrome, and I represent the PHTS community in this ERN. The slogan is share, care, and cure. What is useful for the PHTS community for us? The clinicians, the center of excellence, the laboratory, the data, the foundation, the association, the research group. It's very important, and the best practices. Which benefits and for whom? For the clinicians, for the data entry, for the information, a PHTS literacy only for the PTEN mutation. For the researchers, for us it's important to create the research, the communication with the center of excellence and between the center of excellence on the rare disease, because the rare disease have a lot of comorbidity. So we have it's very interesting to the transversal studies between different clinicians. Cross-border research between USA and Europe or the other states. For the patients, for us it's very important, the clinical guidelines, a unique clinical guidelines as a unique clinical trial, multidisciplinary team, because for a moment maybe there is a fragmentation of information. So it's important for us to unify the informations. For the PHTS community, a new research, a unique platform in which share all the information. It's very important maybe create a PHTS international registry, because if we have a high number of data, maybe it's important for increase the research. The first time, 5 years ago, when we created the association, our dream is to arrive to Cleveland Clinic. And the dream to build a bridge between Europe and USA. And maybe we are arriving to build this bridge. So I'm special thanks to the PHTS families and patients here, the clinicians and the researcher, the Cleveland team, Christine, the PTEN Foundation, absolutely. And I want to thank my association, PTEN Italia, and their families. We are trying to realize a dream. Maybe we try. Thank you for your attention.
Okay, now let's go to PTEN Research and Lab Progress with Dr. Vush Stombulic. Vush is a PhD, is a professor with the Department of Medical Biophysics, University of Toronto, Princess Margaret Cancer Centre, University Health Network, Toronto. God bless you. Ontario, Canada. He runs the Stombulic Lab, which aims to discover, characterize, and target the critical molecular nodes involved in cancer. Dr. Stombulic Particular interest is the biology of PTEN, a major tumor suppressor and one of the most frequently altered genes in human cancer. Utilizing biochemical, molecular biology, and genetic approaches, as well as partnering with experts in the area of structural biology and protein turnover. Moreover, through collaborations with clinic oncologists and direct involvement in several clinical trials, his lab is seeking to extend the mechanistic understanding of cancer pathways towards improving precision therapeutics approaches to cancer treatment. Welcome.
Thank you very much. Let me just find I think we have to close this. I've done this before. Full screen. I'm sorry? Oh, the show, yeah. No. Oh, I thought it was the page display, no? Sorry? Oh, gotcha. Yeah. All righty. Thank you. I'm really happy to be here, and I really want to extend my thanks to Kristen and the foundation for inviting me here to share some of the things that we're doing in the lab. And it's a rare opportunity, actually, for us to connect with the patients. And I'm going to take this opportunity to actually show you some real data from the lab. There's a tendency in communicating with the patients to give overviews and summaries and sort of sugarcoat some of the data and show it in ways that are maybe not as real as what we're seeing. And so I'm going to show you some old stuff, and I'm going to show you some relatively new stuff and sort of give you a bit of a context as to why we're fascinated by these things. So my lab works in Princess Margaret Hospital in Canada, and we're basically a basic science research lab where we try to create situations in animal model systems or cell model systems that are resembling that in patients. In other words, we're trying to create situations in which we can study the disease without actually having to go in the patient. That gives us the flexibility to try out some things that are obviously not possible in patients, and also gives us platforms for drug development. So I think that one of the things I'm going to touch upon here is that how can we still, or how do we still not be able to, how are we not able to explore PTEN deficiency as a therapeutic opportunity is one of the big things that we're thinking about quite a bit. So back in 1998, this is 20 years ago, and it's hard to believe that that is the case. I was working in a fairly large lab of TAC-MAC, who's a mouse geneticist, and we created what was at that time, and it's still considered probably the first model of PTEN deficiency and a Cowden syndrome. At that time, what we knew from Dr. Eng's work was that Cowden syndrome was caused by mutations in a single copy and loss of heterozygosity in some of the other places. So we basically decided to take the gene out in the mouse. So we using knockout technologies, we created mice that are either missing one copy of PTEN or both copies of PTEN. And what became remarkably interesting is that if you delete both copies of PTEN, you would actually lose I don't think, oh yeah, I can see it there. It is embryonically lethal, and with consequences that I'll show you in a minute. But what became even more interesting is a couple years later, when we were able to actually follow the mice that have a single copy deletion for about a year, we noticed that they were developing some of the features that are found in human Cowden's patients. And that was, in fact, and we followed a very large group of female mice because they were easier to handle at that time. And we found that many of them developed breast cancers and many of them developed endometrial cancers. And we've heard before that those are, in fact, some of the hallmark cancer features, if you will, of Cowden syndrome. Our focus is on cancer. I work in a cancer hospital and in a cancer research institute. But some of the things that we're seeing here, and these mice have been used by hundreds of groups since, and some of the things that we're seeing here and some of the things that other people have done with our mice touch on issues of autism and cognitive cognitive dysfunction, touch upon other issues that are associated with the spectrum of disorders. So I promised you data. So here's 1998 data. So these are the embryos. On the left, you have embryos that are effectively Cowden embryos because they're missing one copy of the gene. On the right-hand side, you actually have embryos that are missing both copies. And this black stain that is actually being deposited is a measure of cell division. So the more black you see, the more division there is. So this was the very first hint that, in fact, reducing PTEN activity, which is basically what happens when we genetically delete both copies, increases cell proliferation. This is one of the hallmarks of what happens in cancer. When we took cells out of these animals, and you don't have to look at the left-hand side of this slide, but maybe just on the right, and what you actually see is the cells that are either, again, missing a single copy of PTEN, or two cell lines that are missing two copies of PTEN. If we expose these cells to killing agents, so for instance, poisons or high temperature or osmotic shock, which is basically trying to suck all the water out of the cells, they're actually resistant to these factors. So not only do they proliferate more, not only do they divide more, but they actually survive better. If we now look at the mice that I was mentioning that we followed for a year, you find that we found that endometrial and breast cancers were almost completely penetrant in these animals. So even though in humans it takes far longer for these conditions to develop, growing these laboratory animals for 1 year, which is basically about I would say between a third and a quarter of their natural lifespan would reproduce, in fact, the conditions that we found would take dozens of years in humans to develop. As I said, these mice became models for many things that we have done since and what other people have done as well. And one of the most important consequences of this, and there has been a talk before of everolimus trial. Everolimus hits a downstream effector of what PTEN in fact does, and I'm going to go a little bit into details of what that is. I'm going to tell you sort of a molecular story that is more recent. Everolimus hits something that is downstream, but what it hits, it's in fact a pathway. We call them pathways. These are cascades of molecules that are talking to each other. And it hits a cascade that is called the PI3 kinase signaling cascade. And some of you may have run into this term when you were reading in the literature or what is actually available on the net and some of the brochures. And this is the main pathway in which PTEN participates. And for many, many years, for 20 or 18 to 20 years, we thought that this is in fact the major consequence of PTEN loss. And in fact it is. It remains the major consequence. Because if you look at the tumors from these animals, when you lose PTEN, you see features of activation of this pathway. I'm not going to necessarily point to what this is, and that is reproducible in almost every single cell type in which we can delete PTEN. So with that in mind, if we actually, as I said, we know that PTEN loss leads to hyperactivation of the PI3 kinase pathway. We also know that the targets of this pathway are in fact increased proliferation, increased survival, all these features that we have known before as we were observing our mouse and cells. And these results have not only been done by us, and we, independently of others, I mean, multiple groups have come with these same conclusions using their own models, and those models have been used in thousands of studies in which this has been reproduced to such extent that the measure of PTEN loss is in fact, or the PTEN loss is measured by activation of this pathway. But what is actually incredibly frustrating, and I'm standing here in front of you and saying, we knew this 20 years ago, is that PTEN deficient cancers are not sensitive to PI3 kinase inhibiting therapeutics. And there are therapeutic drugs in the clinic that are hitting this pathway, and they're hitting it pretty well, but they're completely inefficient in PTEN deficient patients, which is completely paradoxical because PTEN loss would be the reason to give these patients those drugs. So there must be something else that is going on. And this has been an incredibly frustrating path that we have all gone down. And I think that Dr. Eng could speak of that, and many others in the room. We've gone down this path really believing in what we have seen in the late 90s and what was reproduced in all those different systems. And that is that PI3 kinase inhibitors may in fact hold therapeutic promise for PTEN deficient patients. So this is how PTEN looks like if you if you put amino acid sequence. It has a N-terminal, or on the left-hand side is the phosphatase domain, on the right-hand side is something that binds lipids and has all these other domains. The way we think about PTEN is also with a lot of decorations. So we're actually looking at various modifications that happen to the PTEN protein, and these modifications are incredibly instructive in how PTEN in fact works. So we're basically focusing on how these modifications are changing PTEN behavior in both the activity of it as an enzyme, because it works as an enzyme, but also localizing it within the cell. And without going into details of what these are, some of them are called phosphorylation, others are called acetylation, some are protein-protein interactions. I'll tell you a little story of what we put together, which may explain the reasons why the patients with PTEN deficiency are not insensitive to PI3 kinase inhibitors. And to tell you this story, I have to introduce a couple more modifications to identify. The one is a very telling modification at the end of PTEN, which linked it to the DNA damage response. This is something that is we all have, and we all are exposed to DNA damage in our cells. And it's also linked it to a mechanism of how it actually gets retained in the nucleus. So you will remember Dr. Eng's yolk in the middle. PTEN actually lives not only in the cytoplasm, but actually lives in the nucleus. And that was pretty fascinating to us for about 10 years. So going very quickly, if you expose the cell that is deficient for PTEN to DNA damage, it starts accumulating these red dots. And those red dots are in fact an indication of damage. So we're able to measure damage. So in the middle of half an hour after damage, these cells accumulate.